Hi guys, welcome to biochemistry lecture. It's Wednesday, October 14th. Can you hear me? Can you hear me well?
Yes, sir.
Yes.
Thank you. Thank you. So we are starting chapter 7. We're going to talk about carbohydrates today. We have 2 lectures on that. Let me open the PowerPoint. Let me open the PowerPoint. And as usual, if you have any questions, please interrupt me. All right. Can you see my screen?
Yes.
Good. All right. So we're going to talk about carbohydrates and also glycobiology. So as you'll find out, carbohydrates play important roles in biological systems. So specifically, we'll talk about our learning goals for this chapter. Will be first, we're going to learn about structures and names of monosaccharides. So, so saccharides actually comes from a Greek word saccharon. Saccharon. Saccharon, which means sugar. Sugar from Greek. And so we'll learn that these sugars can be monosaccharides, okay? Can be small molecules. And these small molecules, they can exist as an open chain or ring forms. So we'll look at both. We're going to learn how to draw them in an open chain, in a ring form. We'll learn how to go from open chain to ring form and back and back. Okay, so then if you take 2 monosaccharides together, you will make a disaccharide. You'll make a disaccharide. So basically these are 2 monosaccharides linked together through some kind of a bond. Can you think of a disaccharide before we even start discussing those? Do you know of a disaccharide that comes to mind?
Is sucrose one?
Sucrose is one, that's right. Do you remember which monosaccharides compose sucrose?
Not quite.
So sucrose, which is a table sugar, right?
Isn't it composed of glucose and fructose?
Yep, glucose and fructose.
Is lactose one also?
Sorry, lactose is another one. Can you think of which 2 monosaccharides compose lactose? Sucrose, glucose,
Is it glucose and
yeah, what's the second one?
galactose or something?
Galactose, that's right. That's right, glucose and galactose. So the interesting thing about sucrose is that both glucose and fructose as sweeteners are much sweeter than sucrose. But you put them together, they become less, the molecule that you make is sucrose, and it's actually less sweet, and that's the one that we use as a table sugar. It's just that it's more available, cheaper, right? And so that's why sucrose is used as a sweetener. All right, so we'll talk about polysaccharides. So these are disaccharides. There are also polysaccharides where you have lots of molecules of these monosaccharides together. Can be hundreds of thousands, can be millions, There's one that is missing. What's between monosaccharides and polysaccharides? Let's say, can you think of a molecule that has 15 monosaccharides? How would you describe it? What kind of saccharide is it? What kind of prefix am I looking for? Starts with an O.
Isn't it oligo or
Oligo. Yep, oligosaccharide. Oligosaccharide. All right. And so finally, we'll learn about biological function of glycoconjugates. So what we'll find out is that, especially these oligosaccharides, they expressed, for example, on various proteins, right? So most proteins actually, when we talk about proteins, something that we did not even cover in our lectures is that most proteins actually are glycosylated, which means they have sugars attached to them. The sugars are also attached to lipids. They attach to cell surfaces, right? So many cells interact with each other through their saccharides, primarily oligosaccharides. And so there's a lot of functions, biological function for these glycoconjugates. All right, so carbohydrates. So named so because they have the formula of certain number of carbons per certain number of molecules of water. And remember, so these carbohydrates, they're the most abundant macromolecules on Earth. And the most abundant, how is it produced? What's this process? Carbon dioxide plus water. Oops, wrong way. This is how we burn it, right? This is how carbohydrates are burned in the body, for example. So what I wanted to actually to ask you, it's actually the other way around. That's why you're being silent, I guess. Here, I'm going this way. So what is this process? So you grab water from soil, you grab carbon dioxide from the atmosphere, and you make carbohydrates. So what's this process? You know that, guys.
Photosynthesis?
Photosynthesis. What do you need for photosynthesis?
Light.
Light. And what else is produced in the during the photosynthesis? Just look at the formula. So we have waters, yeah, and the same number of carbons. What is left out?
Oxygen.
Oxygen. So that's why photosynthesis is so important, because we're going to be producing oxygen. And replenish the atmosphere with oxygen that we use to burn things and to breathe. All right. So produced through photosynthesis in plants. So carbohydrates range from as small as glyceraldehyde, 90 grams per mole, to as large as amylopectin. So for example, this one is 200 million grams per mole. It's a huge molecule. They fulfill a variety of functions. So for example, energy source and energy storage. Can you think of a carbohydrate which is used for energy storage in plants? So,
Starch?
Starch, good. Starch in plants. How about in animals?
Glycogen.
Glycogen. And where is glycogen stored? In the body?
The
Muscles
liver?
and liver.
In the liver, yeah. Is there glycogen in the muscles? I'm not sure. I know the main depot for glycogen is in the liver. That's where it's synthesized, stored. And you know that obviously when you eat sugar, right? Eat candies. So most of it will go immediately into the liver and undergo polymerization, basically from glucose monosaccharide, you will make polysaccharide, which is glycogen, which will be deposited in the liver. And some of it will go through glycolysis, will go and be deposited as lipids in the lipid storing cells, right? Adipocytes. But for example, glucose, you know, that is the main source of nutrient for the brain, right? So for example, when you take a test and you haven't eaten much or you didn't have good breakfast, what will happen? Your brain will starve and your liver will start breaking down glycogen into glucose. And that's the only source of food for the brain, right? So the brain cannot use fatty acids or proteins as a source of food. So, so this is for energy storage. Now, structural component of cell walls and exoskeletons. So we talked about bacterial cell wall, for example. Remember the enzyme that breaks down bacterial cell walls? We just talked about that in chapter 6. Chapter 6, the enzyme that breaks down cell walls in bacteria. No?
Was it cytokines or something? I forgot how to pronounce it.
Lysozyme. Lysozyme, right? So also that obviously it's a structural component in plants, cellulose, right? Cellulose, and you know, for example, we don't have the enzymes that break down cellulose. How do we get the glucose from cellulose? Okay, which animals have the ability to digest cellulose?
Ruminants.
Ruminants, right. And how do they do that?
Fermentation through their rumen, basically, through their four-chambered stomach.
Yeah, so they have microorganisms, right? Some kind of bacteria that express the enzyme known as cellulase. And we will also learn about the reasons why we cannot break down cellulose, but we can break down starch, right? So we can eat potatoes, but we can't eat, I don't know.
We can't eat hay, for example.
We can't eat hay. That's right. We can eat potatoes, but not hay. And also exoskeletons, for example, chitin, right? So chitin would be an example of an exoskeleton. So most bugs will have exoskeletons. If you step on a cockroach, you can hear this characteristic cracking sound. So you just broke the chitin or the exoskeleton of the bug. And so we will also learn that carbohydrates are used in cell-cell signaling. So as I mentioned to you, many cells express these oligosaccharides on the surface, which are designed to recognize other types of cells. And that's very important, for example, for this functioning of the immune system, where the immune cells express these saccharides to find the sites of inflammation or the sites of invasion by a pathogen. All right, and as I mentioned, they can be covalently linked with proteins and lipids. Any questions about this slide? So a lot of, not so much written, but you can see we can be talking about this for the whole day if we want to. All right, so basic nomenclature. So the number of carbon atoms in a carbohydrate plus suffix -ose. So for example, 3 carbons, triose. And so triose has also a prefix. And the prefix is based on the type of the carbonyl group. If it's aldehyde, it's an aldose. If it's a ketone, it's a ketose. Remember aldehydes, right? For aldehydes, we have a carbonyl bound to 1 alkyl group and 1 hydrogen. And for a ketone, carbonyl bound to 2 alkyl groups. So here is the examples of an aldose and ketose. So the simplest aldose is glyceraldehyde. It's an aldotriose. So aldo, let me choose a blue pen. So aldo is based on the aldehyde group, and triose based on the 3 carbons, 1, 2, 3, right? And ose is the prefix for the carbohydrate. Now, why is it glyceraldehyde? Well, remember this molecule. When we talk about lipids, we will see this molecule all over the place. What's this molecule? Anybody remembers the name? It's a 3-hydroxy group alcohol.
Is that a glycerol?
Glycerol, right? So glycerol, the main component of the lipids. If you oxidize one of the hydroxy groups into an aldehyde, then you get glyceraldehyde. That's if you oxidize this one, right? If you oxidize this one, which is the middle one of the glycerol, then you get a molecule known as dihydroxyacetone. So another way to look at this is how this molecule can be derived from acetone. So this is acetone, and there are 2 hydroxy groups, this one and this one. The hydroxyacetone, and it's a keto, right? It's based on a ketone. Trio, 1, 2, 3 carbons, keto triose. Again, ose is a sugar. Okay, so now we know what aldoses are, we know what ketoses are, right? And we know how to name sugars based on the number of carbons. Triose, tetrose, What's after tetros? What's the next one? So tetros is 4 carbons. What's 5 carbons?
Pentose.
What's glucose? What kind of sugar is glucose? How many carbons?
6.
So what kind of sugar is it?
Is it ribose for 5?
No, ribose is 5. Ribose is 5, yeah. But I'm asking about glucose. So how many carbons? So you said 6 carbons in glucose. So what kind of sugar is it? What kind of ose?
Hexose?
Hexose. Hexose is glucose. Yep, and ribose is 5. So we'll see ribose later in this lecture. So we'll talk, so for enantiomers, so we know that enantiomers are stereoisomers, which are non-superimposable mirror images. All right, so here we designate enantiomers on the basis of the chiral center, which is most distant from the carbonyl carbon. And we're going to designate as D or L, just like in our amino acids. And D and L isomers are enantiomers. For example, L and D glucose have the same water solubility. So let me ask you something. So you have L and D glucose, which are enantiomers, right? So they have the same water solubility. Let's say they have the same melting point, right? Do they have the same color? What do you think? Two enantiomers. Do they absorb light equally? So remember, color comes from the light absorption, right? Visible light absorption. Yes, they absorb light equally. So remember, with enantiomers, it's only if you have a plain polarized light, right? They will absorb that differently. But since the light is not, so white light is not polarized, it oscillates in all possible directions. So the enantiomers will absorb that equally. How about taste? So we know that glucose is sweet. Will the taste be different between L-glucose and D-glucose?
I would guess not, because isn't it like only biological systems that can tell the difference between like L and D? So I would think that the taste would be different.
Yes. And remember we talked about chiral drugs? I'm just trying to look up if actually L-glucose sweet at all or not. I need to look this up. I can't find it right away. But so why is that? So why do biological systems differentiate between two enantiomers?
I think it would be like the way that it's like the reception of that molecule. It's looking for just one of those enantiomers.
Yeah, so why are we looking for one of the enantiomers? Why don't biological systems have the same type of interaction with the other enantiomer? Just think of proteins. What are proteins? Racemic mixtures?
No.
Nope.
All of our amino acids have the same
Yes. So proteins are chiral molecules which are present as single enantiomers. right? So now let's say if you have L amino acid, right, in proteins, and when this interacts with L-glucose, now compare, still the same L amino acids now interacting with D-glucose. So what's the stereochemical relationship between these two types of interactions? LL and LD. LL versus LD. In other words, I can ask you, let's say there are two chiral centers in your molecule. There is RR, and the other molecule has RS. What's the stereochemical relationship between them?
They're diastereomers.
Diastereomers, that's right. So, so these are diastereomers. Remember, for those to be enantiomers, you have to invert each stereocenter. So RR would be the enantiomer of SS. But since we inverted one stereocenter but not the other, these will be diastereomers. So these complexes, this one and this one, are diastereomeric. If they're diastereomeric, they have different physical properties. They have different physical properties, they have different taste, they have different everything, different solubilities, right? Even it's possible that binding is different, right? So the L-glucose will bind to some kind of protein, D-glucose will not bind to that protein, and will generate all different properties. All right, good. And so most hexoses in living organisms are D stereoisomers. Although we will find some sugars which occur in the L form, and for example, L-arabinose. So we'll look at that sugar as well. So enantiomers are mirror images. We know that already. So this molecule is glyceraldehyde, if you remember, right? So aldehyde, 2 hydroxy groups. So it's a aldotriose, has still single stereocenter. And obviously these will be non-superimposable, L and D. All right, so based on L and D, so remember, so we're looking at the chiral center, which is the farthest away from the carbonyl group. For glyceraldehyde, it's very easy because there's only one stereocenter. And remember, for glyceraldehyde, if the hydroxy group is on the right, it's D. So in other words, it rotates plane polarized light in the clockwise direction. If it's to the left, it's L. And the reason why we know that, if you recall, when we talked about amino acids, remember when we talked about amino acids and I told you that with amino acids, let's say you have carboxyl group, you have side chain, right? And if you have the amino group on the left in the Fisher projection and hydrogen on the right, this will be L amino acid. And I told you that the reason why we call it L is not necessarily because it will rotate the plane polarized light to the left. It's only because it's based on a structural resemblance of this amino acid with glyceraldehyde, right? So if the hydroxy is on the left, and it's L-glyceraldehyde. And if amino is on the same side, it's L-amino acid. So now we're actually comparing the glyceraldehyde itself, and this is where the amino acid nomenclature comes from. Okay, so also keep in mind the Fisher projection, the way we actually draw them is based on the idea is that the horizontal substituents are towards us and the vertical ones are away from us, right? And so we don't really need, with Fisher projections, we don't need to do these wedges or dashes, right? We can just draw straight lines, and we know that this is a Fisher projection formula, and in carbohydrate chemistry, it's utilized extensively. So if you guys still struggle with Fisher projection, or if you don't remember from OCHEM 1 what that is, I suggest you go back to your organic lecture notes or your textbook and read about that, okay? Or I'm sure you can find a lot of lectures on YouTube that explain Fisher projection formulas.
I have a question.
Yeah.
Just to make sure, so we're looking at the position of the OH to determine L and D?
Uh-huh.
Okay, thank you.
All right, so these two sugars, for example, now these have two chiral centers, right? So there's carbon 1, 2, 3, and 4. So this is erythros, and this is trios. 1, 2, 3, and 4. Erythros and trios. So why are these D? Can you tell me why these are D? What are we looking at? Which carbon are we looking at to determine if these are D?
Carbon 3.
Carbon 3, right. So remember, if you have multiple stereocenters, it's the carbon which is farthest away from the carbonyl group or carbonyl carbon. So it's carbon 3 and the hydroxyl is on the right. So these are D sugars. But so the stereochemistry at carbon 3 is the same, but stereochemistry at carbon 2 is different. And so these kind of isomers are known as epimers. So epimers are stereoisomers that differ at only one chiral center, right? So in other words, if you have 10 chiral centers and one of them is different in two molecules, then these two molecules will be diastereomers, first of all, but secondly, the particular kind of diastereomers, they will be epimers. If there's more than one carbon that is different, then these will not be epimers anymore. And they obviously not mirror images because they're not enantiomers. So they will have different physical properties, water solubility, melting temperature. And so trios, shown here, is a C2 epimer of erythros. So when you say that's an epimer, you usually indicate at which position it's an epimer. And here it's a C2 epimer because the difference in stereochemistry is on carbon 2. It's a C2 epimer. And they both, the sugars, have the same orientation around the last carbon in the chain. All right, so these are tetroses, right? 4 carbons.
So those second carbons, they don't rotate. They can't rotate.
How do you want to rotate them? So these are Fisher projection formulas. And in the Fisher projection formula, the way you will build it, first you put your aldehyde, right? Then you put your carbon 4 on the end. And then you put your 2 remaining carbons in the middle. Like so. So rotate, you mean switch the hydrogen and OH? Switch their positions?
Yeah, they don't rotate.
Oh, it's not going to be rotation. It's going to be, the only way you can, these can swap is if you break bonds.
Oh, okay, okay.
Yeah, you cannot exchange them by a simple rotation. You have to break this bond. You have to break this bond. It's just like going from R to S. All right. So diastereomers, stereoisomers that not mirror images have different physical properties. So these are, so erythros and trios are diastereomers. So we just talked about that. So let's skip the slide. Now, more about epimers. More about epimers. So let's look at the epimers with pentoses, or sorry, hexoses. So the main type of hexose for us is D-glucose. Again, how do we know that it's D? What carbon are we looking at?
5?
Sorry, which one?
5? Is that
5.
right?
Yes, 5. We're looking at carbon 5, hydroxyls on the right. It's D-glucose, correct. Besides this chiral carbon, there are 3 other chiral stereocenters, right? And so let's look at mannose. Let's look at mannose. And we see carbon 5 still has the same stereochemistry, which means it's going to be D-mannose. But let's look at the other stereocenters. Carbon 4 is the same. Carbon 3 is the same. Carbon 2, on the other hand, this has different stereochemistry. So of the 4 stereocenters in glucose, 1 stereocenter has been inverted. Therefore, mannose and glucose are C2 epimers. C2 epimers. Now, glucose and galactose. Again, galactose is a D sugar here, carbon 5. We're looking at the other stereocenters and we find that carbon 4 stereochemistry is different. So glucose and galactose are C4 epimers. C4 epimers. What about mannose and galactose? What's the relationship between these two? Are these 2 epimers as well?
No, I think, can't they only differ at one location to be epimers?
That's right. So here they're different at 2 locations, yes? And so 2 out of 4, which means that they're diastereomers. I mean, they all of these are diastereomers, but epimers is a particular case of diastereomers where only 1 location is different. Diastereomers. All right. So keep in mind, mannose and galactose, a good thing to remember. Now, speaking of which, on the test, what may happen is I will give you a list of various sugars that I want you to know the structures of. So, so you can see there are a lot of rules and there are a lot of structures in this chapter, right? If the previous chapter was more math based, this chapter is more about structure and in a way kind of memorization of the structure. But for example, I will not ask you to memorize the structure of galactose. But what you can do, you can memorize the structure of glucose and then go from there. So glucose, you know that galactose is C4 epimer of glucose. So you draw the structure of glucose and then invert carbon 4. Same with mannose. So you know that mannose is C2 epimer of glucose. Draw the structure of glucose and invert at position 2. Right, so there are many tricks that you can use, and I'll be mentioning those as we go through the material. But in general, what I'll do sometime next week, I will give you, or not next week, maybe even, well, yeah, maybe Monday, Tuesday. I'll give you a list of structures or carbohydrates that I want you to know the structures of. Okay. So that you know, since there are so many of them, obviously I don't want you to memorize every single one of them. So common carbohydrates in biochemistry. So ribose, so we just mentioned that. Standard 5-carbon sugar. Where is ribose found?
RNA.
RNA. Why not DNA?
DNA has deoxyribose.
Deoxyribose. Good job. So glucose, galactose, mannose, fructose is the ketose form of glucose. Okay. Okay, so we just looked at that. So aldoses, so these are 5-carbon aldoses. Here's the D-ribose. Now again, note the stereochemistry of all these carbons which are away from the carbonyl. These are all these sugars. Arabinose, xylose, lysose. Yeah, I don't know how to pronounce this one. Lysose. Lysose. So now aldoses with 6 carbons, whole bunch of them. Again, I will not ask you to memorize each one of them, but I will want you to know these 3. Glucose, mannose, galactose. So you can see that there is allose, altrose. So altrose is the epimer of allose. This hydroxy on the right, this hydroxy on the left. All the other ones are on the same side, right? Glucose, mannose, these 2. Gulose, I don't know how to pronounce that one. Idose, galactose, talose. All right, many different possibilities for different stereocenters. And obviously many of these are present in various biological systems, right? So in animals, we primarily utilize some of the main ones, glucose, mannose, galactose, but in other biological systems, various other carbohydrates are utilized. So all of these are important for life on Earth. I don't know why this slide keeps, maybe I copied and pasted it several times in different places. Okay, so ketoses. Now, so these are 5-carbon ketoses. So this is ribulose. Again, it's a D sugar. But the important thing is that now the carbonyl group is a ketone, right? So it moved from the primary carbon, sorry, from the terminal carbon to the second carbon here. There's xylulose. Here there's a ketone as well. And it's a D. It's a D one. And that's the fructose. This is the component of sugar. So fructose, surprise, surprise, is a ketose, right? and it's also D-fructose. D-glucose and D-fructose are components of sugar. Sorbose, tagatose, all ketoses. All right. So these are all open chain forms of sugars. Now we're gonna switch to cyclic forms and look at that in detail as well, because cyclic forms are the most important for their biological function. So one thing to so things to keep in mind. So aldehyde and ketone carbons are electrophilic, and alcohol oxygen is nucleophilic. And so when aldehydes are attacked by alcohols, hemiacetals form. When ketones are attacked by alcohols, hemiketals form. So, and these reactions form the basis for cyclization of sugars. So remember this reaction from Ochem, was this Ochem 2? Ochem 2, I think. Ochem 2. When we talk about the reactions of aldehydes and ketones. So here, the alcohol will attack the aldehyde carbonyl. This goes here. And usually for this reaction, a proton would be required. Okay, I will just abbreviate this without the proton, but you get the idea. So when this attacks here, then you form a molecule known as a hemiacetal, right? So you remove the carbonyl, carbonyl is no longer there, but you introduced the alcohol. Now there's a new bond, carbon-oxygen bond shown here. Now it's important to keep in mind that this is not a oxidation reduction reaction, right? This carbon had 2 bonds to the oxygen, and it still has 2 bonds towards the oxygens. So the oxidation state of this carbon has not changed. And then the second alcohol can come in, right? And replace, so let's say you protonate this now, H+, turn this into a good leaving group, and this can attack this carbon, and water will leave. Water will leave, and this will give you acetal. So now there are 2 molecules of alcohol are bound to this carbon that was part of the carbonyl group here. So this is transformation between aldehydes and acetals, and the molecule of water is released. Now the same can be observed with ketones. Same idea, 1 molecule of alcohol will add. You will form hemiketal as opposed to hemiacetal. Add second molecule, and you form a full ketal. So ketone goes to ketal, aldehyde goes to acetal. So you guys remember these reactions? So it still would be useful for you to look at the mechanism, even though I kind of drew the basic features of the mechanism. But usually these reactions occur in the presence of acid catalyst. They do not occur in the presence of base catalyst. You need an acid catalyst. And an important thing here is that these reactions are reversible. You can see there's reversibility sign or reversibility arrow, right? So aldehydes can go to acetals, and acetals can decompose to aldehydes, which means that the sugars can go back and forth as well. All right. So now let's look at the same reaction only with monosaccharides. So here's nucleophilic alcohol. Nucleophilic alcohol will attack the electrophilic carbonyl group. So what we need to do here is we need to be able to convert this Fischer projection into, no, I forgot what it's called. There's a name for it. So it's a 6-member, so basically ring form of the sugar. Ring form of the sugar, you just have to convert one into the other, basically keeping an eye on all the stereocenters. So this is something that you have to practice. Basically, kind of remember the way you look at the Fischer projection is carbon 2, H and OH towards you. You look at carbon 2, H and OH towards you. Carbon 3, OH on the left, H on the right. Carbon 3, OH on the left, H on the right. You kind of follow this ring and keep looking at each stereocenter, and that will help you determine if the OH is up or down, up or down. If you go this way or if you go back, this will help you to determine if the OH is on the left or the OH is on the right. Now, okay, so these are called Haworth or Haworth. Haworth formulas. Now I intentionally put a bunch of such problem in your sapling homework, so you keep practicing that. Okay, and just one more thing about sapling, guys. Sapling is not that many points, so if you lose points by doing sapling problems, that's okay. Just think about it. It's you only get 10 points for the entire homework. You get 100 points for the whole exam, for one exam, right? So even if you lose a few points doing sapling, that's just nothing. So use sapling not as a way kind of to get the points that you need. Use sapling as a learning tool because it really walks you through the solution, right? And I think here going between the open chain and Haworth formulas will be a good exercise. And
Can you explain what happens at carbon 5? Because it seems to be like counterintuitive.
yeah, carbon 5 is the most difficult one because we switched the chain. So carbon 5, what you will need to do for carbon 5, you will need to have carbon 6. You will need to have carbon 6 in this position, right? Just 1 second. So let's redraw this. Let's redraw this. Like so. OH. OH. H. H. Carbon 4. OH down. H up. So for carbon 5, what we're gonna do, I'm going to rotate that so that the carbon 6 will take the position of the oxygen. Oxygen will take the position of the carbon, of the hydrogen, and hydrogen will take the position of the of carbon 6. Okay. So did you understand what I just did? Basically, I did the rotation for carbon 5 about carbon 4 and 5 single bond. Okay. And the reason why I did that is so that I can match this now with the Fisher projection. And so now I'm going to start walking down this Howard formula. So I'm starting at the carbonyl. My first stop is on the carbon 2, right? What do I see? I see OH on the right, H on the left. Now imagine you lying down kind of, and your head is sort of horizontal, not vertical anymore, right? You're kind of lying down and you're looking at this molecule in such a way that these two bonds are away from you and H and OH are towards you, right? And what do you see? You see OH on the right, H on the left, and that's what we see. Okay, we'll go to carbon 3. Carbon 3, now OH on the left, H on the right. That's what we see. We'll go to carbon 4. OH on the right, H on the left. That's what we see. We'll go to carbon 5. The same thing. OH on the right, H on the left, and then CH2OH. Does this make sense?
Yes, sir, it does.
Yeah, I know it's something that you have to just do several times before you get it. So
You'll want to rotate that carbon 5 about that single bond between carbon 4 and carbon 5, so that you can align the OH with the carbonyl.
So you align the CH2OH with the carbonyl, yes. Mm-hmm.
Okay. Yeah. Okay.
Because ultimately it's this chain from the carbonyl to CH2OH, which is on the vertical axis of your Fisher projection formula. Okay. Yeah, keep trying, Sapling, and come to my office hours. We can do it together. If you, you want to share screen, what you've done, I can look at what you've done and we can discuss. All right. So now, once we have done all of this, now this OH and the carbonyl are in a position for nucleophilic attack. So we're going to form a hemiacetal. Now, the difference in sugars is that we're forming here a stereocenter. We're forming a new stereocenter, and the new stereocenter can be such that OH is on top in this structure, or the OH is on the bottom in this structure. So and so this orientation of the alcohol around the carbon, this one, it says here is variable and transient, right? So it can be on top, can be on the bottom, but keep in mind, you have the equilibrium sign. So in other words, if you made this compound, which is, we will learn for reasons a little later, it's called beta D-glucopyranose. Whole bunch of new words for us. Beta D-glucopyranose. But what's important is to realize that if the OH is on top, it's beta. But it can go back to the open form, and the open form can recyclize again with the OH being on the bottom. And now it's alpha D-glucopyranose. Okay, so we'll talk about this phenomenon in a moment. So this carbon here, C1, is a key carbon in carbohydrate chemistry because it's the most reactive carbon in sugars. So it's a new chiral center once it cyclizes, and we call this carbon anomeric carbon. So this anomeric carbon, when the former carbonyl oxygen becomes a hydroxy group, in other words, the carbon becomes an anomeric carbon, the position of this group determines if the anomer is alpha or beta. Okay, alpha or beta. So the anomer then, so this is beta anomer. This is alpha anomer. Alpha anomer. So if the hydroxy group is on the opposite side trans of the ring as the CH2OH moiety, the configuration is alpha. Let's go back. Here, we are looking at, let me change the color so we don't have everything in blue. Let me use red this time. We're looking at this, carbon 6, and we're looking at OH. These are opposite sides, right? CH2OH is on top of the ring, OH is on the bottom. Okay, so this is opposite. So it's alpha. You may have guessed. Here, CH2OH and OH are the same side. So we're going to call these as beta anomers. Beta anomers, cis. Okay, so as I mentioned to you, lots of rules here to remember. That's why practicing is the best way to do it, and keep working on your sapling problems, guys. All right, so you may already notice that, well, first of all, it's D also. Let me tell you a trick. Let me tell you a trick. This was D-glucose, and we knew that. Why? Because of the C5 carbon, right? And now look, if the C5 carbon gives rise to D-glucose, what happens to C6 carbon? It's on top, right? In other words, if this was L, in other words, if the OH was on the left, where would C6 carbon be? If the OH was on the left,
On the bottom.
On the bottom, instead of this hydrogen. So the hydrogen and the carbon 6 would have switched the positions, right? And so you don't really need to draw. So in other words, you can predict if you have D sugar from both Fisher projection formulas and Haworth formulas. From Fisher, your OH has to be on the right. From Haworth, the CH2OH, the 6th carbon, has to be on top. If the 6th carbon is on top, that's a D sugar. So this is beta because the OH and the C6 carbon on the same side. It's D because the C6 carbon is on top. It's gluco because it's glucose. And what is pyranose? We're going to here. And it's pyranose because of these heterocycles. And this heterocycle is known as pyran. And this one is known as furan. So pyran is a heterocycle that has, sorry guys, I should have turned off my Skype, which I didn't. I apologize. Let me quickly do that. Share screen. Okay, are we back? Can you see the slides?
Yes.
Okay. So so this is a pyran, and so because you have a 6-member trin with the oxygen, so therefore the sugar that is similar to it is known as pyranose, right? It's a 6-member trin with the oxygen. Now, if you have a 5-member trin, we will see with fructose, for example, that will be furanose. And so those will be based on the sugar furan. And so the anomeric carbon is usually drawn on the right of this side. Okay. All right, so let's look at some examples. So what are we looking at? So first of all, we see that this is glucose, right? So it's good to recognize glucose from both the Fisher projection formulas and Haworth projection and Haworth formulas. In Haworth, I would suggest to you that now this can change. In this particular case, it's alpha. For some reason. Okay. This can change because it's anomeric position. But your carbon C2 hydroxyl is down, carbon C3 hydroxyl is up, carbon C4 hydroxyl is down, so they switch positions, and carbon 5 CH2OH is on top because it's a D sugar, right? So if you see something like this, this will be glucose. Don't worry about anomeric position. Anomeric position, the OH can be on top, can be on the bottom. If it's on the bottom, you can see it's on the opposite side of the CH2OH, which means it's alpha. CH2OH on top, it's D. It's glucose, and it's pyranose because it's a 6-member trin with the oxygen inside. Now, this is beta D glucopyranose because the OH and CH2OH are on the same side. Beta D glucopyranose, and that's the structure of the pyran. Now, this is furanose, so this is fructose based on the structure of furan. And so in the fructose, what do we have here? Again, we're looking at the anomeric position. So the OH group is down and CH2OH is up. So this will be opposite. Therefore it's alpha. Now it's going to be D because CH2OH is up. It's fructo because it's fructose. This is something that the more times you actually do problems with fructose, the easier it will be to remember the structure. And this is beta. This is alpha, this is beta. Beta is because the OH and CH2OH are on the same side. And that's furan. Okay, guys, do you have any questions so far? I know there's a lot of material. But it's all pretty simple, as I mentioned. But once you go over this a few times, there should be no problem. Okay, so in the remaining time, what I would like to do is maybe go over these two slides. Now, maybe just one slide. Let me do just one slide. So obviously, these monosaccharides, since they're 6-member trains, they will adopt the chair, mostly chair, not boat. Yes, boat is not good. So in most cases, chair, and there are multiple chair conformations possible, but require energy for interconversion. And so in most cases, the chair conformation for glucopyranols that you're going to draw is this one. And you can see here there is, if you compare these two, like why is this one on the left more favorable than the one on the right? Tell me.
Is it because of sterics?
On sterics, so what kind of sterics are we talking about here?
So the CH2OH and the OH on the right side, they're close to each other, and they'll want to be farther apart.
Okay. Is it just one particular bad interaction, or there's more?
I guess like all of them are interacting with another like large group.
Yeah. So if you look at it, all of these are what? They are equatorial. 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 Remember, equatorial is much more favorable. Equatorial is much more favorable. And there's a big one, also equatorial. And that one, equatorial. These ones, on the other hand, are all axial. And if you look at these two, just like you said, the two axial ones, the reason why axial substituents are unfavorable is because what's known as 1,3, continue, 1,3 don't remember, okay. 1,3 diaxial, 1,3 diaxial interaction. So it's sterics, that's correct. So very unfavorable. And so sugars, when they adopt the cyclic form, then it's very important to draw them in the most favorable orientation, because that's what they're gonna look like in biological systems, and their properties will be based obviously on the type of ring form that they will adopt. All right, guys, do you have any questions about today's lecture? Lots of new material, right? Lots of new material. Looks like a lot of things to perhaps memorize, or perhaps derive some sort of a tool which will allow you to to put things in the context and reduce the amount of memorization. Just like I told you with the mannose and galactose. If somebody asks me to draw a structure of galactose, what I will do, I will draw glucose and then switch the position of the OH at carbon 4. That's how I would normally do it, because I put it in the context. I don't memorize the structure of galactose, but I do know the starting point, glucose. So, all right, so the homework should be available in just a few minutes if you want to start early. And I will see you on Monday. Have a good weekend guys.
Bye. Thank you.
Mm-hmm.